no matter how you feel about chemistry. We got to get started. Okay, today we're going to talk about puberty. And in talking about puberty, we have one of a famous set of questions that two of which we will deal with in this course. What comes first, the chicken or the egg? Okay, and whichever side you come down on, doesn't matter. But it's of the same sort of a question as whether the fetus or the mother determines time of what? What did we talk about where that question came up? Birth or parturition. This one is the timing of puberty. And I'm going to go through a number of slides today that begin to address that question, but we need to consider what goes into it. It turns out not to be simply a question of age by any means. So, if I were to ask you this last question, what do you think? What triggers the timing of maturation of the gonadal axis? Is it development of the gonad, or is it the development of the hypothalamus and the pituitary? Pardon?
Is it the latter?
You'd say the latter, based on what?
The hormones that would be necessarily secreted.
The hormones that would be necessarily secreted. But aren't they being made all the time?
Before the formation.
Beforehand? Okay. What do you think comes into it? Triggering puberty. A combination of both. Okay. Anybody else want to weigh in? Suggestions? Okay. Well, let's get started. Let's start with this slide. For the term ends, I'm going to figure out which one of those it is. Okay. There was a crucial set of experiments that was done in the late 80s, in the late 80s and 70s and early 80s. And it was a series of samplings of plasma LH and steroidal hormones taken from subjects, and these happen to be female subjects. They also did it with males. I believe this was done in Scotland, or the United Kingdom. But they had a series of individuals who had been born agonadal. They didn't have gonads at birth for whatever reason. And there were obviously some normal controls. And what they found was very, very interesting. First of all, this is plasma LH. Okay, birth is down here, and you'd expect plasma LH to be low. And it turns out to be low in normal females, but somewhat higher in agonadal females. And then as you go along, this stays low or even goes a little bit lower until about age 10 or 11, at which time it goes up and begins to follow an adult pattern. But notice what happens in the agonadal subjects. They have this same decline, or have a decline, somewhat similar to what you see in the normal subjects. It's fairly shallow though, and it zooms way up after this timing. But these are agonadal. So can they go through a true puberty?
No,
Why not?
if they don't have the target that's going to respond to the signal.
Okay, they don't have the gonads, in this case the ovaries, to respond to the LH or FSH signals. What does this also say about the development in these individuals? Notice the level is high here, it's also high here. What's missing? Or what perhaps isn't missing? Maybe that's a better way to put it. Why are the levels so high? Here and here. Somebody other than Jenny.
Absence of negative feedback.
Pardon?
Absence of negative feedback.
Absence of negative feedback. Okay, if there's an absence of negative feedback, no gonads, you don't have any steroids feeding back to suppress production of gonadotropins, what does this also say about the functionality of the hypothalamus and the gonadotropes? There's a limit, or how about the fact that they're functioning at all? That data says they have a functional hypothalamus, functioning gonadotropes. They are responding as you would expect them to respond in the absence of a negative feedback. And there is something going on that is independent of the gonad at about the time of normal puberty. Let's talk about that. Let's talk about some of what's potentially going on. Because the pattern in agonadal males looks pretty much the same. And I might add that these levels here are only seen in normal subjects at the time of menopause. So, in males, and it turns out that there's a somewhat similar characteristic pattern in females, it depends on when you look during development as to what the pulse rate of LH-RH and subsequently LH is coming from, going into circulation. So prepubertal, during this childhood stage, you're only seeing small peaks of LH a few times during the day, not very close together. In this early pubertal phase, which also coincides with the maturation of the adrenal cortex, so it begins to make weak androgens and as well as glucocorticoids in higher levels than it did during childhood. So you've got some steroids being produced by the adrenal down in this age. They actually can go down as low as about age 8. So right in this region, the adrenal is maturing. The adrenal axis is maturing as well. And you start seeing in early puberty, over here, you start seeing the pulse rate of the LH, what's called the LH generator, increasing, and you're starting to see the amplitudes increase, particularly at night. In mid-puberty, this pattern continues to be magnified. You see increased pulse amplitude and frequency even during the day, as well as during the night. And then finally, as an adult in the post-pubertal period out here, the pulse frequencies in males are going to run between 1 per 90 to 120 minutes, both night and day. So you lose the day-night contrast in the secretion pattern. Interestingly, if I starve this male, I make him into a long-distance runner as a child or early adult, I can delay this maturation. If I have an individual who demonstrates anorexia, if they start over here, they delay this maturation pattern. If they've already gotten to the adult stage, when that condition starts, so whether it's natural or whether it's psychological, self-induced, what you find is a reversal of the pattern. So this pattern starts to show day-night differences and ultimately give you a low pulse rate with a low amplitude. It's as if you reversed puberty at the hypothalamic level. So we know there's enough information in the literature now to indicate that this is driven in part by the energetics, what's that? By the energetic status of the individual and by the energetic status of the individual and the rate at which they're growing. Get down the list a little bit. All right. This slide overlays some of findings that I and other colleagues have had on top of a theory that's been around since the early 1970s on how puberty actually occurs. And it's called the gonadostat theory of regulation of LH control and timing of puberty. And what Brumbach and his colleagues did was to look at how sensitive the hypothalamus was to steroidal feedback. And they did that in a variety of animal models and also looked at it by passive observations in humans. And what they found was that as a fetus matures, it becomes more and more sensitive to negative feedback. That may correspond to a change in the number of steroid receptors in the neurons that are driving, for instance, the kisspeptin production and or LHRH. Then as you go through infancy and childhood, this is they're claiming is a very sensitive period for negative feedback. But as you begin to transition toward puberty, what you see is an increase in the release of GnRH, I just showed you that, as well as an increase in the responsiveness to GnRH. So the pituitary increases the numbers of receptors for LHRH, GnRH, and you get increasing levels of LH for every pulse of LHRH. you're getting increased gonadal responsiveness of gonadotropins to rising sex steroid levels. So you're having receptor numbers and the numbers of cells that express those receptors increasing in the gonads during this time period. And you're starting to see this sleep-associated increase of LH. And then finally, full activation in the female of positive feedback for LH during cycles. Well, this pattern was a nice hand-waving description of the data that had been collected by the early 70s. And you know, it tried to give you some idea. There's change in steroid receptors, negative feedback, increase of the GnRH and the gonadotropins as a result of some of these receptor changes, probably cellular changes as well, and then taking you through toward the adult. Well, there was a lot of conversation in the community in the 80s particularly about the need for a certain amount of fat to allow puberty to go forward, or what happens when you change the energetic status of somebody by making them exercise, finding them as demonstrating anorexia nervosa, being in conditions of starvation. Any of those things are changes in the availability of energy to all systems in the body, but particularly the reproductive system. And remember, the reproductive system is one of those that can be shut down without damaging the viability of the individual. It's not good for the species, but it can preserve function for the individual. Well, when we had been doing some work, particularly in New Guinea, we had seen very late ages of puberty, close to the early 20s, actually late teens, early 20s in females, and into the mid-20s in males. So there was questions about, okay, what's going on here? What's holding things up? We found out there's probably some nutritionally related things, particularly iodine intake. I've gone over some of that before. I'll probably review a little bit of it again. But in addition, what we found is that one of the indices that turns out to be linked to thyroid function as well as to steroid function is sex hormone binding globulin. It's made in the liver. Thyroid hormone stimulates it as well as estrogen levels stimulate it. And it has this life pattern of falling from middle of pregnancy through childhood, and then coming up during this pubertal period, rising to a peak, and then falling back down once you've gotten to adulthood, and then there's a bump later on in the aging population. Well, the significance of sex hormone binding globulin is that it is what determines the free steroid levels in circulation. So the testosterone, the estrogens bind to this binding globulin. And if you've got a lot of this, the free sex steroid levels are low. If you've got a little of this, they go up. So the free sex steroid levels should follow essentially the mirror image of this. So what's the implication? Notice how this looks relative to that? It's the same pattern. Even down to here. There may be some other things going on over here, but you're through puberty at this point. So what our suggestion is that part of the mechanism for the gonadostat is actually a change in the circulating levels of sex hormone binding globulin. which alter free steroids in circulation. So, we know Grumbach published in the 70s this pattern for the gonadostat, but they didn't really describe fully what the mechanism was for it, and we're trying to suggest that may well be part of the mechanism. Yeah. Now I want to go to this one. This was an interesting study published in the late 90s, which was based on histology of both the rat and ultimately of the human as well, looking for the number of Leydig cells and whether they retain dependence on LH. Under various conditions, various ages, and what the functions were. And intriguingly, what you find is that the number of Leydig cells increases. That's not a big surprise during fetal stages, and their growth is independent of LH. Once you get to late stages of pregnancy, birth is at 21 days in the rat, you start to see a shift toward production of testosterone, and it's all LH dependent. In the human, we've got this LH analog in circulation, chorionic gonadotropin, coming largely from the placenta. And depending on fetal stage, you're seeing a lot of it or a little of it. Okay, at 78 weeks, you get the onset of Leydig cell differentiation and the onset of testosterone synthesis, and it's dependent, this functionality. And remember, what's significant about that stage, 7 to 8 weeks?
Sex differentiation takes place.
Pardon?
Sex differentiation.
That's at the very beginning of sex differentiation. Okay. So this very early stage of sex differentiation, you're seeing the Leydig cells responding to LH and circulating HCG. Okay. As fetal age goes on, you start to see LH synthesis in the fetal compartment and its effects on the testis via the receptors, the LH receptors, which are also developing. You get LH in plasma, and by the end of the first trimester, you're hitting the highest levels of HCG, and you're getting maximum testosterone synthesis from the available Leydig cells. So this is all HCG dependent. Then, like the rat, later on, here we are out at the second trimester to the end of pregnancy, you're seeing the maximum LH in fetal plasma, and you're starting to see LH and HCG in plasma decline. The Leydig cell number, looks what it's doing. It's going down. And testosterone in plasma follows that. So you've got a change in the numbers of Leydig cells functioning, and notice that this is the stage during which you're getting internal phenotypic and external phenotypic changes in that individual. And this is just taking some of that and actually getting it down to the actual numbers of Leydig cells and showing you that in the fetal compartment there are mature Leydig cells, stationary Leydig cells, and involuting Leydig cells, depending on how long into the pregnancy you look. And then postnatally, this is interesting. Look at the numbers of identifiable Leydig cells prepubertal. Does that jar anyone? Does that bear any significance there? There's lots of them in the adult after puberty. What about during childhood? What does that say? What does that say about the status of testosterone production in the male gonad during childhood? It's not very high. Why isn't it very high?
The number of Leydig cells is
Not
there.
Okay, because the Leydig cells, if they are there at all, are not identifiable as such. They're either reverting to an earlier cell stage, more like a stem cell, where they're not producing a lot of androgen, a lot of testosterone, or they've gone away and they're being replaced later on by a second group of Leydig cells that were derived from some stem cell pool. But there's a nadir in Leydig cell function during childhood. Okay. You don't, in the female, you've got follicles that may be present, and if they start developing, they fail. Remember that? How many of them fail in the ovary prior to ovulation? Can anybody give me a number or percentage of oocytes that fail to develop fully prior to puberty? 100%, okay? They all fail. They start to differentiate toward growing follicles, toward the normal maturation you'd see in oocyte development. Because the hormones aren't there to support them, the cyclical hormones aren't there to support them, they all fail. And if you go back to that slide that gave you numbers, it's thousands and thousands and hundreds of thousands of oocytes that actually fail during that period of time. Okay. Once you get to maturation, there was an investigator in England called Tanner who did some real basic work on cohorts of children. And what he did was he measured their length and he measured their weight. And then he developed, as time went along, he developed scales, semi-quantitative scales, to try and assess how these individuals were passing through puberty. And in the case of the male, he looked at the length of the penis, he looked at the development of pubic hair, and he looked at the percentage of individuals, or the frequency of the testis being larger than 4 millimeters in volume. He also developed some interesting interesting tools in order to do this. How would you figure out that a testis was more than 4 millimeters in size without removing it from the individual? Any guesses? Pardon? You could measure the scrotum. You're chuckling there. Any guesses? Come on. You've got to be more imaginative than that. Yeah.
Displacement.
Pardon? Displacement? Displacement? Okay. So it's one possibility. What he actually did is they made up a series of kind of ovoid or egg-shaped spheres out of silastic plastic. Okay. And he had them built so that they were of varying sizes, varying volumes. And then he matched them. And that was a goniometer, or gonadometer, I should say. Now, in the female, the markers were breast development to the stage of the area around the areola and the nipple becoming raised relative to the chest wall. He also looked at pubic hair development in the female, and he looked at the initiation of menses, that is the timing of the first menses in these individuals. Now, what do you notice as there's a real characteristic shift in terms of the age in females versus males. What do you notice in terms of the first signs of development in females? Yeah, how many years earlier? 2 to 3 years earlier. So this breast development is leading increase in testis volume or penis length by a couple of years. The timing we think of for puberty, I mean the classical one for females is the initiation of menses, the first menstrual period. And that's occurring down here on average somewhere between 11 and 13 years. The pubic hair development is very close in timing between both genders. What's that being driven by? Pubic hair or hair development, particularly pubic hair, but also works on the axillary hair and facial hair. What drives that? Estrogens? It's androgens. And it's not just testosterone. In matter of fact, that index is probably at least substantially supported not just by testosterone, but by the androgens from the adrenal gland. So you've got changes in the adrenal leading this charge through puberty. And you've got females developing responses a couple of years earlier than males. If everybody were to stand up and we measure everybody and we put a histogram up, what would that histogram look like for males and females? Would they be the exact same thing?
No.
Pardon?
Height?
Height. Statural height.
No.
What would they look like?
They might both look normal, but the men would be the higher peak.
Okay. If you were to do that measurement, what you usually find in a population is that both are distributed, it's not that bad, both are distributed normally, this is female, and the males are skewed toward being taller. This is frequency, and this is height. Now, part of that height attainment is determined by the effects of growth hormones and steroids on the ends of the along bones where growth actually occurs. That's the source of statural growth. It's the growth of the long bones from the ends. When estrogen levels are high enough, you will get a closure of the epiphyseal plate. You'll get a calcification of that area of the bone, which halts statural growth. So, given that information, let's look at this area of the female curve. Can you predict anything about that subpopulation of females other than their height? What did I just tell you about the attained height? What helped determine it?
They'll be tall.
Pardon? Yeah, but they'll be tall. But why will they be tall? Pardon?
The epiphyseal plates will continue to grow
Okay, they'll grow until what?
until the estrogen levels, until the puberty ends, right?
Okay, so what's going to be true of this population of females, say, relative to this group?
later.
Okay, these are probably mature, late matures.
Or does it mean their puberty is just longer?
Yep. Or prolonged puberty. Okay. If there are populations that are going to favor one of those or the others, and so you can go out and find populations with high means for stature, okay? High average statures. And when you do, you often see not necessarily nutritional, but you see timing of puberty being late. Okay? I'm thinking particularly of experiences with Nilotic people, a group of nomads, pastoralists in northwest Kenya, and they are very, on average, very tall, but their maturation is also very, very late. On the other hand, the work in New Guinea was with people who had average stature, which is very much stunted relative to Western norms. Now, the other thing that Tanner provided a picture for trying to estimate where people were developmentally. Okay? And it's been used in lots and lots of places. The caveat on Tanner's results is that they were done on Northern Europeans. Okay? Now, do Northern Europeans look and behave exactly like every other people on Earth? Okay, how's their nutrition level? It's good now, and it's been good for a long time. Certainly back into the 40s and 50s when Tanner was taking this data. Okay. So the problem is that they're genetically, they aren't really diverse, and they have had a high nutritional claim for a long period of time. So how applicable is this if I go to New Guinea and try and evaluate development in highland peoples?
Not very.
Or sub-Saharan Africa? Okay. So the question comes up, what is normal, okay? What is normal development? What's the normal timing for things? There is a wonderful book that any of you who are going into medicine should read. It's called Normal at Any Cost. And it is a book about growth hormone and recombinant growth hormone. Growth hormone was isolated from pituitaries in the 1950s and 60s. So there would be groups that would take cadaver pituitaries, grind them up, isolate a whole variety of proteins, but one of them was growth hormone. Now, what use was that growth hormone that you could think of possibly? What would we use that growth hormone for? Human growth hormone.
You mean modern?
Pardon?
In modern time?
Well, I mean, modern times since 1950.
Don't they use it to like make livestock produce more eggs and milk and?
Not human growth hormone. That's bovine growth hormone. And okay, what about human growth hormone? Anybody around here who could use a bit of human growth hormone?
Oh, is it for steroids for sports?
Well, that's a later advent.
Do they use it for height?
Height for whom?
For people?
For whom?
For children?
Okay, people are children born and demonstrating low statural growth. So if you looked at their growth pattern, they're in the below the norm.
Wait, not even people with like legal dwarfism?
Pardon?
Not even like legal dwarfs, just people who are kind of short?
Well, you start with shortness, then you test for whether or not they're producing growth hormone. Okay. And now whether producing several other hormones as well. But the point is that when this was initially being done, they were looking for individuals who were demonstrating abnormally slow or low growth rates. And under those circumstances, they would inject isolated human growth hormone. Okay? And they found that it helped increase the rate of growth toward the norm. Ran into some problems in the late 60s and early 70s because people were demonstrating that in certain human populations and certainly in cattle and sheep, there were these things called prions. Does anybody know what a prion is? Okay. They're a protein-based material that seems to have the ability to replicate or to propagate. Okay. It's diseases like Jacob-Monod and scrapie in sheep and cattle. But they're viral diseases. And if you're taking your material from cadavers and using the kinds of preparatory procedures that have been used for growth hormone up to the 60s, you have the potential of carrying prions into that preparation. Later on, it was the HIV virus that was potentially a contaminant. It wasn't really until the 80s that they had isolated the genes for human growth hormone. And when they did, they then had the ability to insert these into cells that would overproduce growth hormone. They could then harvest that from a non-cadaver source, so it wouldn't be contaminated with viruses or prions. And you had a very ample source of growth hormone. You could treat everybody who potentially needed it as short stature. But the manufacturers also had a lot more available. And when you've got a lot of something available, what happens to the price? It goes down, okay? So what's the other way to increase a price of a product that you have a lot of? Pardon?
Increased demand.
Increased demand. How do you increase demand for a hormone like growth hormone?
Convince everyone that they need it.
Pardon?
Convince everyone that they need it.
Convince everyone that they need it. Like how?
Telling you not in the right perception.
They say it's good for you, by saying it helps you with certain
They increase the normal height.
Okay, so if somebody still is in the normal range, but wants to be taller, okay? So it's voluntary, giving it for voluntary purposes, or using it in athletics to enhance muscle building. The point was that there was an attempt at that point to change the definition of normal to allow use of growth hormone in more places. You can tell me at some other point whether you think that's ethical. Okay. So that's the story about growth hormone and growth. And there are Tanner came up with a whole series of other staging keys, and this is a plot of age versus the timing of each of these stages. And females faster than males, and usually more condensed. Males tend to be a longer period of time. Okay. But again, this was normal for Northern Europeans. Whether it's normal for all populations worldwide is an open question. Okay. Now we talked about sex steroid binding globulin a little bit already, and I think I can leave it at that. What time have we got? 48? Okay. Next time, we're going to get into thyroid hormone functionality. So by the time you get to your turkey next Thursday, you're going to know all about thyroid hormone. We've gone through puberty.